Hello, in this video we will discuss about the inosine monophosphate IMP to generate the AMP and GMP. Inosine monophosphate is basically also known as the IMP is the central metabolic molecule which that is used for the synthesis of DNA, synthesis of the other things, the synthesis of the nitrogenous bases for DNA replication, this replication of the cell division, mitosis. So the cell signaling molecule we will also learn in this video, and basically it is the reciprocal regulation is the GTP for needed for AMP synthesis and ATP needed for GMP synthesis. So anyhow, the inosinate IMP. Inosinate, what is the function of the inosinate we will learn in this video. So the inosinate is also known as the inosine monophosphate, which that's contain two ring containing nitrogenous base. This is the nitrogenous base. And in this way, the glucose is the fructose is basically the ribose sugar, not fructose but ribose sugar, which that attach with the carbon number 5, the phosphate group. So anyway, this is the inosinate. It will be go the further pathway is the two type. First we will discuss, and later on we will discuss another path. So this is the aspartate will be entered, and in this way the adenylosuccinate synthase enzyme used, and the GTP will be used to produce the GDP and inorganic phosphate group release. And anyhow, in this way here is the formation is the basically inosine monophosphate aspartate formation, and in this way the oxygen side will bind with the aspartate. So the fumarate will be released after the aspartate entering. The fumarate will be released in the presence of the adenylosuccinate lyase enzyme. The adenylosuccinate lyase enzyme used, and in this way the adenylate AMP. Adenylate AMP is basically the AMP which that will be formed. Remember, this AMP will be converted into the cyclic AMP. We will learn during the cell signaling pathway. So here is the inosinate will enter by the hydrolysis to IMP dehydrogenase enzyme to produce the NADH from the NAD plus to produce the xanthylate, the XMP. This xanthylate XMP is the GIN and the glutamate, and the absorption of the water. After the absorption of the water, the ATP will be used to produce the AMP, and the pyrophosphate will be released. And XMP glutamate, the XMP glutamine amidotransferase enzyme used. And in this way, the guanylate GMP. This guanylate will be formed. This is known as the GMP, the guanosine monophosphate. It is also known as like the adenosine monophosphate. So this is basically the two molecule will be formed. Remember that things. This molecule we will understand about in during the cell signaling pathway. So the when ATP decrease, the AMP increase. When AMP will be increased, it means the second messenger is the cyclic AMP will be formed. And on the other hand, when GTP decrease, the GMP increase. The ratio of the GMP will be increased. Anyhow, I hope you make sense about the biochemistry of the AMP and GMP formation. So now we will discuss about the cell signaling pathway. The cell signaling pathway means that any ligand can bind with the receptor, the G couple protein receptor we are taking in this video. So the primary messenger, the ligand is the primary messenger with a bind with the extracellular domain. An intracellular domain contains a trimeric protein alpha, beta and gamma, which that here is the increase of the glucose or decrease of the glucose will regulate the signaling pathway, and it can be epinephrine, glucagon, insulin or other things. So this is the mitochondria. In this way, the activation through GTP to convert the GDP, and the GDP containing alpha subunit will be released by the activation containing the alpha GTP. This alpha GTP will be activate the adenylate cyclase enzyme. In the presence of adenylate cyclase enzyme, the ATP will be used to convert the cyclic AMP, and the pyrophosphate will be released. The two inorganic phosphate group will be released, remember, and this will become a secondary messenger, will activate the other proteins like protein kinase A phosphorylation. The cascade of other phosphorylation will trigger the DNA, and this DNA triggering mechanism will transcribe the gene, and each gene are present in the DNA, and the gene on DNA which that transcribe into the messenger RNA. So after this messenger RNA, the transcription will go into the cytoplasm from the nuclear pore to outside into the cytoplasm to synthesis of that protein, and this protein is the response. So that is why in this video we have discussed about the inosine monophosphate to generate the AMP and GMP, and the cell signaling pathway, how it will be work. So the cyclic AMP is conversion due to the adenylate cyclase after the production of the AMP. So the AMP simply will be converted through a adenylate cyclase enzyme, so it is become a secondary messenger intracellular, while the extracellular is the ligand, for example epinephrine, glucagon, insulin, or other hormone. So thanks for watching. If you like this video, please make sure to subscribe, like, and share, and ask the question in the comment below for understanding better. So I hope you like this video. If you like, please thumbs up and also share. Next video we will learn about another topic about the biochemistry lecture. So bye.